buddy. So this will create problems for us somewhat in the potato programs, because we all want to go out at the pre-plant or at planting application. Chitosan's got to stay out of the phosphates, some of the nitrogen forms, the sulfur, any of the negative charges. We have to stay out of that tank mix, because we'll glob onto and make a mess in the bottom of the tank. There's a few products we have found across the country with some of our dealers. We've been able to subvert this with some phosphate chelators. When we all get back to Idaho, I will be doing some playing here with the redox products to see if this is possible with theirs too. I suspect that we will be able to tank mix with their products here with us. The other one, so cationic, no negatives in the tank. The other one that is absolutely critical is the pH buffering. Anytime you're playing with this, the solution in the spray tank has to be at a 5 pH or below. If you can't do that, don't put it in the tank. It is naturally one of the aspects of chitosan that as you move up the pH, as we get more basic with this, it starts to flocculate. There's actually applications in other industries where they want it to do this. In the oil drilling, fracking industry, they will actually use chitosan to plug off some of their cracks if they're losing the drilling mud out of fissure. One of their techniques is they'll send chitosan down with a pig and then a caustic substance right behind it. Send the chitosan in, they hit it with the high pH, and it instantly starts to flocculate and web. Now they can start pumping other chemicals in behind this to bind to it and seal this crack off, rather than trying to fill the whole thing full of mud or other expensive products. So it's just naturally the way chitosan is. You've got to absolutely, the spray solution has to be at a 5 or below to keep it in suspension and in true solution form. Anti-transpirant. Influences the pathways containing jasmonic acid. Jasmonates exhibit some activity similar to the plant hormone abscisic acid. That is one of the responsible for signaling for stomatal closure. I realize I don't have enough experience yet. I don't know what this is going to do for maturity in corn. This is one of the questions I get asked frequently here in the last years we get around the Midwest. I don't know if what this is going to do to increase or decrease maturity in corn. The stimulation of lignin production helps to strengthen stomatal guard cells. So as we are creating a better stock here, that lignification is actually happening on the stomatal guard cells too, and making them stronger. The first thing that's going to happen when you make an application is it's going to trick the plant. The stomata is going to close right off. They've done a lot of studies. If you'll Google the transpiration effects of this, a lot of work has been done in this department, because we're all trying to conserve water, other than this last year for some people. But they will do these studies on pepper plants because they transpire so heavily. So they've actually proven this time and again, that they can reduce the transpiration rate in pepper plants by about 40%, 40–45%. So not quite half of the transpiration rate we can cut down with the pepper plants. After that, those stomatal guard cells get stronger and actually makes it able to pump a little easier. If you get into a heavy rainfall, so the plant's got wet feet and it's trying to get rid of moisture, it can actually work to open that stomata further to help the plant get rid of the moisture faster. Forms a vapor barrier. It'll actually seal coat the plant. This chitosan naturally forms into films. There's some other aspects of this. Our military, the bandages that they give our troops when they go out in harm's way on the front line, those bandages are infused with chitosan, because it is such a powerful antimicrobial agent for the bad guys that it prevents any infection. Number two, it's a very powerful coagulant. So any bleeding that goes on, we keep a jug, a spray bottle in our shop at the farm all the time now, just for nicks, cuts, scrapes, burns. It will actually bind to some degree. It will bind to your pain receptors, so you get a burn. I don't usually promote this, but I will actually mix a 2% solution, put it in a spray bottle, and my children will ask for this when they get a sunburn, because it will actually take the edge, take the pain off of sunburns. So films. What's that? Oh yeah, yeah, yeah. Freeze tolerance. So again, stimulating the stomatal closure helps retain water within the cell structure, aids in the movement of nutrients within the plant, both xylem and phloem mobile. When your plant moves into this, like it knows what's good, like it knows how to do this. It knows it's trying to stay alive. It knows what to move and where to move it and what it needs to do. The chitosan just facilitates those actions and those movements. Works against pathogens that will promote ice crystal formation. They call these ice nucleating bacteria. And this Pseudomonas syringae is kind of the most prolific one for that. They will actually use this microbe at ski hills when they're manufacturing snow. They will actually add this in because it's going to start forming the ice crystals sooner. This organism will be on your leaf surface. Its entire goal in life is to make your plant cells rupture. That's when it gets to eat. It will actually work to start nucleating, forming ice crystals at several degrees above freezing. So 34, 35 degrees, you will have these microbes on your leaf surface working against you to try to promote freezing in your plant. And there's some studies, again, if you look this up, that the chitosan's actually been very effective against these ice nucleating bacterias. Wetting sticking, chitosan naturally forms into films. There's an electrostatic charge that occurs. So this being a very, again, cationic molecule in nature, your plant will naturally, the soil naturally will have a negative charge. So when we apply the chitosan onto that, there's an electrostatic bond that happens that glues it to the leaf surface. Anything that you mixed with it you got in the solution is now stuck there as well. There is a research paper you can find if you dig hard enough. One of the big chemical companies funded a doctorate research study in United Kingdom a year ago, 2 years ago. They took a strobilurin in chemistry, and they were doing this in a controlled greenhouse environment. They sprayed the strobe on just by itself on one side. They took the strobe, just used a generic chitosan, mixed them together, on the other side. And then they proceeded to give this 10 inches of simulated rainfall. And at the end of this, they went through, I think it was radiospectrometry that they were identifying this with, where they had put the strobilurin out straight. They had less than 10% left on the leaf, in the leaf. They could only find 10% of what they put on. Where they had mixed the strobilurin with the chitosan, they were still greater than 90% retention of the strobe there. So it is very effective at delivering. The medical side of this, when you start Googling chitosan adjuvants, everything you find will be medical because they talk about an adjuvant as a delivery mechanism. So they take medical grade stuff here and they will actually put their other molecules onto this and they will use chitosan as the delivery mechanism to get it into your body. If we were a group of scientists standing around here, we wouldn't be calling this chitosan, we would be calling it polyglucosamine. Same stuff you take for your joints. We just took a glucosamine and added a whole bunch more of them and created a polyglucosamine. This compound is completely ubiquitous in nature. It is found everywhere. It's here. We just figured out how to harness this and put it to work. There's a couple other constituent components that we will use back and forth in some of our products. First one is yucca shitigera. The other one is quiaya. Quiaya is a soapbark tree, comes out of Chile. Yucca is it is a sterol-based molecule, which actually comes into a play with some of our potato scab diseases. Some of these depends on your scab's kind of a broad term, but some of these, it will be more of a protozoal type life form that causes the infection. Not really a cell structure there at all to speak of that we can work on. But we have seen dramatic reductions in scab. I did a small scale test a couple years ago dramatic reductions in scab. Couldn't understand why, because on paper we shouldn't be affecting this as much as we did. Come to find out that those type of organisms, their cell structure will be more of a cholesterol base. When we apply the yucca, being a sterol-based molecule, actually binds with the cholesterol in these organisms, and at their point of cell division, we interfere with that, with the yucca, and it basically does the same thing that the chitosan does for some of these. The chiaya, that is a triterpene-based molecule. We've actually been doing some testing. We have a new product that we have brought to market now that is a combination of these components here that we've seen some really promising results. I'm going to do some more testing in Idaho this year. We're looking wireworm, armyworm and alfalfa, those type of organisms. We're looking at taking this and spiking our chitosan with it to give us the complete control that we're after. We've had results, we've had some good results. I want complete results, so they're pretty powerful on their own. As a food preservative, again, this film, of this ability that chitosan has to form films and spread and coat and cover, this is only, that aspect just gets made better with the yucca and the chiaya. I've got guys, we've been put on seed potato country, peroxyacetic paracetic acid. They were spraying onto the tubers going into storage to try to help with that fusarium that causes dry rot. We actually, they tell me we've mixed fine with that product and the chitosan, so now you've got a twofold, threefold aspect going on because we carry that around the entire tuber. We will make that entire tuber wet and we will put that chitosan shrink wrap around it on there. So you got the peroxide effects of that, you got the chitosan effects of that. It will actually control the oxygen in that environment. In a seed treatment scenario, we've actually, again, responding to the market and customer input as we've went along, we've gotten to the point that we're actually now, by this spring, we'll have seed treatment label available. As a seed treatment, it's phenomenal. Soybeans, I got slides here we'll talk about in a little bit, but it will actually imbibe inside the seed coat. As long as the chitosan is the first compound that's put on the seed, it will actually get inside the seed coat. And in soybeans, it actually creates this semi-permeable or semi-osmotic environment in there that it will allow moisture in, but if you're planting in dry conditions, it doesn't allow the soil to wick the moisture back out again. As well as, as it comes in contact, any pathogen load that may be present on your seed, it takes care of that. We'll get some residual, a couple weeks down the road with a standard seed treatment rate. Some pictures. Pictures are always fun. Not sure where the pointer is on this one. If you look at this one down here at the lower left, the way you're facing, the two of those on the right-hand side, I was up my dry farm. This is off my own place. This was a combination. I had 2 ounces of O2YS in combination with a biological seed treatment I was working on. This is hard red spring wheat, grown dry land, eastern Idaho. I was just digging for moisture one day, was curious how popcorn fart dry it was getting, and was shocked at the mass of roots that I had in these. So then I got curious. I'd ran out of seed, almost done that year, and didn't have time. We just got a little bin run stuff out and finished up on the field there. So the two on the right are the chitosan and the biological combination. The next one over is absolutely no seed treatment whatsoever. The one on the far left is a conventional pink seed treatment. And if you look at that, there's not a lot of difference between the pink seed treatment and the absolutely no seed treatment. You know, it's kind of the black necrotic tissue working up on the stems where we've done this on with the combination. The stems are completely white. There's nothing, if I remember right, this was 6 bushel. I believe this was 6 bushel difference. Here you can see a difference between the pink seed treatment and the biologicals. The one in the lower center here, this was out of eastern Idaho. I was actually marking spud rows for this guy, and we just got talking about this and that and what I do and what we could do. So anyway, we took a pint per acre of O2YS and put in the mark out. So this was in furrow, pre-plant. That is the only difference between these. So this is the one where we put it on, this is the grower standard. It's a little bit deceiving because he had the seed piece still attached to this one. I had nothing to do with this dig. The farmer found this, sent me the pictures, and was absolutely blown away that how is this even possible. This one wound up, there's actually a little bit of a write-up on this one in the brochures. We got an extra 0 in there. This didn't cause a $4,000 an acre increase, but we did see, he figured about a 20 sack increase right to the line. He's a pretty loyal customer now. The Rockstar. This is over western Idaho in some white edible beans. Pretty obvious to see the difference here where we had one pint of O2 versus grower standard. This was over western Idaho also. It doesn't show so well in this, but this was the treated side and untreated side, and the color difference was remarkable in these sugar beets where they had put the one. Sugar beets are kind of fun to play with this way. If you get sugar beets that are a little peaked or tepid, not the right color green, the chitosan alone, again with the photosynthetic amplification that we will trigger with this, we'll do it, mix this with a little magnesium sulfate in the mix, and you can pull off a parlor trick. It will, in 24 hours, you will see exactly where you missed with the sprayer because the green color will be that obvious.
It's also a 2-point Brix increase.
Yeah. Generally speaking, we will always see a bump in Brix. PPE, both pictures here, we're killing nematodes. So the one on the left, this is actually my brother. This is actually the tractor I usually drive. Tractor's for sale, by the way, if anybody needs a good 9530T. So to throw that in. Yours truly over here. Either way, this would have been a metam application because we didn't ever do telone with the markers, but minimum PPE. It is so safe to work with any of our products that literally long sleeves and pants is all that's required. I didn't usually worry too much about washing my hands before I ate a sandwich in the middle of the day, but I feel better about it now when I'm just dealing with this stuff. Smaller treated, untreated. These pictures are out of Mark's neighborhood. This was off a rather large tomato farm in southern Georgia. Again, the antiviral aspects of this, so think along the lines of PVY and potatoes. This farm, this block, they were infected with tomato leaf curl virus, and this is a kiss of death. There's nothing you can do. It's a viral infection. They don't have anything to treat this with. We came in, started a treatment every 7 days? Once a week. With a pint quart?
Pint to pint. Pint of O2 and pint of nematode.
Okay. So pint to pint, nemasan and O2 once a week. This was the difference. This road you see here is actually in between this block and this block. They're not taken on the other sides of the county. They're taken on the other side of the road. There was a bigger block. It was basically being written off. There was nothing. We had a distributor stepped in, told them they think we can do this. Made a full harvest on this side. Zero is what they harvested off the untreated side. So yeah, we do some stuff for viruses. Peanuts. A lot of this is attributed to nematode control. The difference in the roots here, reniform is the big nematode that we have a problem with down there, and we have done fantastic against reniform. Wheat out of Montana. This was a co-application. This was made in conjunction with a 5-way micronutrient package that this dealer carried. Not really but you look at the difference in the head size here, and whatever we triggered or the stress that we helped alleviate within that crop that was able to allow it to pronounce a heavier reproductive manifestation. This is off the same farm, same products combination. Take a minute to look at that. They just plucked the little boy or stood the little boy out in the middle of the field to show the difference. I forget now, I will find out what the tonnage difference was. This was barley that they had planted as a hay crop, and the farmer had been out spraying the rest of the farm. And when he got back in, he had a little bit of stuff left over, so he just figured he'd run the sprayer out on this. And again, this one came completely unsolicited. The results were that surprising that sometimes.
They don't mix words.
Yeah, this is off a colony, one of the colonies up to Montana. So not a lot of BS with those guys. They have no problem telling you exactly where you stand with them. So again, the soybean seed, you can see some of the growth stimulation effects that we have had it here. Coated, uncoated. This picture here kind of depicts what I was talking about earlier about imbibing into the seed coat, that we're actually going to move in there. We're going to strengthen that. This semi-osmotic event that happens when we get in there holds the moisture in, doesn't let the moisture out. Again, these pictures, when you start Googling, which I hope every single person here will start Googling, because I might be lying to you. I might have made this whole damn thing up today. So everybody go Google, make sure I'm not lying. But these pictures will show up. These are pretty common on the internet. You will see these time and time again as you start researching around. As a food preservative, this was a fun one. My daughter last year in the 3rd grade wanted to be in the science fair. And I'm not that science fairy. Like, I know a little bit about one thing. So we figured out a science fair with chitosan. So we went to town. This is, anyway, 1 potato here, 1 apple. This is 2 different strawberries we cut in half. So I tried to keep things as even, equal as we could with this. We put everything in water. We washed everything. And then on our treated side is on the left here. We just dipped these in a 2% solution with chitosan. So it's mostly water. Acidify this a little, put a little chitosan in. We use the 01 with the OMRI certification on it. Instantly, the potato guys generally jump right on this. And the pictures I have on my phone are a little more, anyway, you can see it better. But we all know what happens when we cut a potato in half, expose it to the atmosphere, and how quick that starts to turn black. This first photo was taken 24 hours after we started this. And within a few minutes you start to get the discoloration going on here. Even in 24 hours you can see this darker ring forming up here. We're still pretty white-like colored with this one. Again, because we've stopped any pathogen growth, we completely encapsulated these seed pieces with this chitosan coat. We've controlled that environment now. No nothing's getting in there. We've stopped the oxygen from getting in. As our experiment went on your right side over here, this is 21 days later we ran this for to see what happened. Our apples actually dehydrated so fast on this side that they cracked open in just a few days. They split in half. Our treated side, we shriveled up, obviously. We never did crack and split. The raspberries, not a huge difference between side by side. The blueberries, however, the non-treated had significantly more shrinkage. I was a little disappointed with this because I fully expected, we wrote in the hypothesis teaching my daughter the scientific method that we expect mold and yucky stuff, and it never showed up. And I couldn't figure out why until we got through with this and went to wash up my wife's white towel and the smell of Clorox hit me. So I think I jinxed this from the get-go that I used one of my wife's beautiful white towels, and that prevented any mold growth from growing on anything. But you'll be happy to know, first place at the science fair. We even beat the 4th graders, so happy to say I can beat a 4th grader. A microbial stimulant. Have you guys you familiar with this new microbiometer? Has anybody played with this? It's a company out of the Carolinas somewhere. I highly encourage all of you to go look this up. That's how you spell it right, microbiometer. This is a device, you go take a little soil sample. It takes you about 5 minutes to do the mixing on the tailgate of your pickup. You let it sit for 10 minutes. You take an extraction off this, put on the little scorecard. Anyway, in 10 minutes, 20 minutes, you will have a numerical value. They will give you this. So right here we had a 458 and a 737. Just got a phone call the other day. They're actually measuring micrograms of biology per gram of soil. So you have no distinction of whether this is bacterially dominated, fungally dominated. We don't know what it is. We just are able to measure the biology here. So on this field, this was actually Nemasan. I'll have some pictures, in fact, right here. So half of this pivot was in spuds last year. This side was in grain. We treated this portion of it with Nemasan, untreated here. So the 458 came off the untreated side. The 737 came off where we had applied 1 pint of Nemasan with 1 pint of O2YS. We were able to clock a 61% increase in microbial activity. So this is becoming it's addicting. If you guys get one of these, I'll warn you, it's a horrible addiction. It is so fun to be able to go make an application with a product like ours like Kevin's, Jason's, like it is so fun to be able to monitor now what's going on in the soil. And literally you do this it will cost you $10 a test. Do it on the back tailgate of your pickup. You'll have 30 minutes total invested in it, and you'll be able to see if I'm full of crap again. You can actually go make an application, do a side-by-side, and see if we did anything for your microbial population or not. So I put this one in. This is a long story. I realize that makes you all sigh because this is long anyway. Phytophthora erythroseptica pink rot. So the farmer owns this pivot here, and he owns this wiper pivot. He picked up the lease on this field last January. The history on this field had been in barley the last 3 years, and it had been in alfalfa for 5 years prior to that. So this is a fairly old photo. The site of this field, this was fumigated in the fall of 2016. It's been general practice in this neighborhood that 1 shot of telone will get you 2 crops of potatoes on a 2-year rotation. So fall of 16, it was in potatoes here in 17, in cereal grains in 18, back into potatoes in 2019. So he picked up the lease on this, decided to put this one into potatoes. Has not been fumigated, touched anything for at least 8 years, 9 years. That we knew for sure nothing had went in there. This field, he was worried about nematodes, so we ran Nemasan and O2 once a month all summer long religiously. We started at planting every 30 days we ran. There's actually a reason why we can pick the timing dealing with GDUs and nematodes, but we won't get into that. But this field, so it got a lot of chitosan this summer, all right? This field, we did nothing because of the fumigation that went on. Got a picture. This is these fields. Well, a little stress event, the 18th of June. They know in this neighborhood, this organism is so proliferant that you get something like this happens, your season better run perfect after this. Because any hiccup at all, your petioles crash, anything, this is a recipe for pink rot right here when something like this happens. So again, the one field picture was taken down here in this corner of it. This is where that event happened. This harvest, when the farmer was to cut to the chase here, this site here that we had ran the Nemasan on, he had 0 claims at the fresh shed. These were Norcotas. This was an early dig. He was pushing onto the early market, running right out of the field. 0 claims, 0 pink rot, 0 nematodes. They did not find a single nematode-infected potato the whole time they ran this one. This field up here, they tried and tried and tried to run this one through the fresh shed, and they couldn't. The pink rot was so bad on this side of the pivot that they couldn't take it. The few loads right here out of the middle, it was a little bit less, so the local shed tried to run a few loads for him. I was talking to the guy at the warehouse, says that they probably shouldn't have ever brought those in. Not only with the pink rot situation they had, but they were running about 90% corky ring spot, which is caused by stubby root knot nematodes. And even if you just have stubby, that's not that bad. But in this neighborhood, the stubby root knots are infected with the tobacco rattle virus. They're the vector for this. That's what causes the corky ring spot. So this farmer's pretty ecstatic. Fumigation now, telones, I'm sure you guys are well aware, telone in my neighborhood's 19 to 21 bucks a gallon. When we did this, so you're $400 an acre on a 20-gallon application. That was pretty standard, 15 to 25. The only time we ever went as low as 15 is if we were doing an over-under and we were putting metam with that. So we'd have 15 gallons of telone, 25 gallons of metam. Start doing the math on this stuff, even a straight metam application, you're starting off in the $300 an acre range. We ran all season on this for less than $100, and they found 0 nematode potatoes on this one. So I'll show you this one. This is down the road, same neighborhood. This is the far south end of it. This summer, this field here was in potatoes. This one was in potatoes. This is the story on your handouts with the frost event. I put this in so that you guys can see that I didn't go take these pictures from this was, literally this is a full pivot and this is a wiper pivot right on the edge of the desert there. He splits these into 3 fields. This side got a quart of O2YS at planting. This side was grower standard. And that story, I say, kind of kind of see the story, or you can read the story there, but this is the side-by-side. So grower standard and 1 quart of O2YS in the markout just prior to planting. That was the difference we saw. When we were out looking at it, this literally, I mean, as bad as these pictures look here side by side, this is an average of both sides. I didn't go pick the biggest one. The farmer was with me, in fact. We didn't pick the biggest one off the good side and the smallest one off the bad side. This literally is an average representation of what those side by side fields, of what that looked like. So again, the stress, or the frost tolerance in this, the one thing I'll caution you on, we've had numerous dealer reports back with this, that when you make a chitosan application, we know that this will run for about 25 days. And they can actually, with some of the testing that's been done, it'll run sort of a bell curve. That the plant activity and efficacy ramps up and then it falls off the other side pretty quickly. So we know 25 days is about where we need to be looking to retreat before that curve crashes and we catch this going up again. However, anytime you have a stress event like this, the clock resets. So if you applied it today and you get froze in 5 days, you're going to come through that one really good shape. If it freezes again in another 5 days, this one's going to smoke you. We don't understand the plant physiology yet, exactly what's going on or how we're being consumed in the plant, but we have learned from the School of Hard Knocks that we get burned up in the plant during these stress events like this. So even if you're running a 25, 30-day schedule here for nematode, something or another, you go through something big and hairy gnarly like this, the clock resets. We need to look at a retreatment again to keep the protection going. Stripe rust and cereal grains. This field was horribly infected. I took this picture 24 hours prior. These were all live viable pustules. This was an ancient grain variety the guy was playing with, and it was as bad of an infection as I've ever seen. We did one application just to see. I gave him this one actually just because I wanted to see what we would do on stripe rust, and we get a lot of phone calls. Anyway, we've done very, very well putting this in for stripe rust control. Longleaf pine out of Mr. Nichols' area down there. This is kind of shows some of the plant eliciting effects. This is off a humongous tree operation down there. Long story, but they're very, very impressed with us. Again, seed treatment aspects of this. They were treating the seed, put 2 seeds in a pot to make sure that at least 1 seed would always germinate so they didn't have empty pots. I can't remember now the thousands of dollars in labor they had to spend to come back through and pluck 1 tree out of all of these plots and transplant because they all germinated. 90-some-odd% of all their pots wound up with 2 trees growing out of them. They were not that happy right at the moment, but they're pretty happy now. We're part of their program now for all of their...
That farm grows 68 million trees a year.
68 million trees a year. Wow.
Yeah.
That's a lot of Christmas. The chitosan, this one stands here alone. Again, the acidification aspects of this is critical. If you can't acidify it, don't put it in the tank. So we actually brought this product to market. This is all it is. There's no secret here. This is 100% pure citric acid. And this has been the easiest way to attack or work with this pH situation.